So it's a pleasure to be here. Thank you for inviting me and getting a chance to discuss a few of the things we're doing with mammalian synthetic biology. So my name is Ron Weiss. I'm from MIT and been working in synthetic biology in general for now about 20 years with a focus on mammalian synthetic biology for about 12 years or so. And so this notion of being able to write full human genomes is really quite exciting to us. And so what could we do with that? So some of the things you've probably been thinking about or hearing about are these notions of, for example, cancer-resistant cells. Can we create customized immune cells? Can we create infection-resistant cells? So it's exciting to think about those kinds of possibilities. Those types of cells really they have multicellular interactions. In some sense, those are individual cells. At another level is this notion of actually getting coherent tissues in terms of being able to benefit from this human genome project, right? And so some of the things that we're interested in pursuing include programmable organoids. I'll talk briefly about what that means. And then beginning to think about this notion of renewable anti-aging tissues or rewritable tissues. So things that you can actually modify the genomes on the fly to try to create new kinds of functions and capabilities, potentially even in vivo. And so what is the notion of programmable organoids? This is something we're beginning to explore. This notion that we can take induced pluripotency stem cells, reprogram them, edit them, put new genetic circuits in them that allow them to proliferate and then make all kinds of, for example, 2D or 3-dimensional patterns by design, and then that is used as a mechanism to create kind of desired tissue, either mimicking exactly normal developmental pathways, or maybe even trying to create some kind of variance of those. And so for that, you really need to be able to have precise spatial, temporal, and cell type specific control. Why do we want to be able to do this? Organoids are really exciting for drug development, disease modeling purposes, also for organ transplantation. So here's a quick example. We're able to take iPS cells, genetically engineer them to make GATA6 at different levels. And so we're able to precisely choose the distribution of GATA6 in this population, low GATA6, high GATA6, and something in the middle. And what happens is that the low GATA6 ends up becoming ectoderm, and then the high GATA6 is mesoderm and endoderm, and the medium GATA6 cells end up either becoming ectoderm or mesoderm or endoderm, depending on their neighbors. And so we get this initial homogeneous population of cells that ends up doing symmetry breaking and differentiating into these 3 different germ layers. So the nice thing about these germ layers is that you should be able to derive any cell type from those, from the endoderm, mesoderm, or ectoderm. And in fact, what we did is we let this system continue to develop, and we're able to notice that it developed and produced a large variety of different cell types. And in fact, what we ended up getting is essentially a liver bud next to brain, and this liver actually had liver functionality. So that was pretty exciting to us. We then took this 2D environment and moved it to a 3D situation where we grew these now organoids, something called a hanging drop, and that allowed them to develop. And we had a very patient graduate student who let these things grow for 2.5 months. And by that time, these things, well, first of all, we're highly vascularized. So they're completely deeply vascularized, allowing nutrients to really penetrate this entire organoid. And that's essentially what you're seeing on the bottom left there. And as a result of that, nutrients were able to reach all the cells, and these organoids became really quite huge. So we're kind of calling them meganoids. So if you're looking over here, this is basically large enough to probably play hacky sack with or something like that. But they're about 100 times larger than anything that we know of in terms of liver organoids. And you really can begin to think about, certainly about disease modeling, but also for transplantation purposes. And so we'd like to move forward on these notion of programmable organoids to enhance those organoids, and also to think about the notion of renewable anti-aging tissues and rewritable organoids as well, where we can add new functionality. So we'd like to create, first of all, certainly many different genetic variants. So that that's pretty obvious, and that's something that we could highly benefit from a human genome project. Right on that, we'd like to be able to create mechanisms that sense everything that's going on there genetically, control everything that's going on, create different kinds of 3-dimensional shapes. And also importantly, being able to dynamically rewrite the genome. So do this in an efficient way, but also to actual tissues. So that really doesn't allow us to use the existing technologies for basically reconfiguring what the human genome is in each one of these cells. So we can add functionality or remove functionality as these tissues are operating. And so for that, we need building blocks. We need synthetic biology building blocks. And we need platforms that allow us to do this notion of dynamic rewrite. And so very quickly, I'm not going to obviously go into the details of these things, just to show you, been able to make some progress on these building blocks. So we've been able to create large libraries of transcription factors so we can regulate lots of activities and create platforms for fairly sophisticated, hopefully, genetic circuits. We have been able to create sensors for many of the things inside the cell. So for example, all the high confidence microRNAs, we can now have genetically encoded sensors for that can then plug into our circuits. We have a platform that might allow us to sense any protein inside the cell and then connect that again to circuits. DNA, there's work on that, mRNA and so on and so on. So those are the sensors, and also be able to engineer cell-cell interactions through either long-range cell-cell communication with this apple-based flavonoid called Phloretin, which diffuses quickly, or something that's really short-range with synthetic notch. And in addition to communication, we have the ability to have physical interactions via coherence. So this is a movie that we took of these cells that we are now programming to interact with one another and then create these three-dimensional shapes by design, hopefully leading to tissues. And so what we really need to be able to do these kind of aspirational applications, again, is this notion of in situ or in vivo genome dynamic write platform. And I think that actually builds up very nicely on the landing pad work that we heard about from Leslie and from Farron, the work that he's talking about with the oligos to change small places in the genome. And I'll talk about that in the next two slides. So we've been able to create these landing pad cell lines that have actually fairly nice long-term expression, about 3 months of good continuous expression from these multiple landing pads. We'd like to have not just 3 of them, we'd like to be able to have hundreds of them that we can integrate to many different places in the genome, very large fragments. So we've been able to integrate 60 kb into each one of those landing pads, and it works really quite well. And we can do these things, multiple things at a time. And for those of you that have background in electrical engineering or computer engineering, you know about FPGAs, field programmable gate arrays. And so as computer nerds, we get excited about those things. And so we like to have this notion where the genome already contains the possibilities of reconfiguration. And what we do is then we provide these small signals, for example, with the ways that Farron was talking about with small oligos, very small signals that we can provide to the tissue. It will get everywhere in the tissue, and those signals will then direct the rewrite in the genome itself, essentially rewiring these genomes on the fly and hopefully allowing us to be able to reprogram function on the fly. And so we have some very initial ideas about how to do these BioFPGAs, if you will. So with that, I'll stop. Lots of people to thank in my lab, lots of interactions outside the lab. So it's been really exciting to be part of the mammalian synthetic biology community, and I'd like to thank you as well. How's it? Timekeeper is okay? Any questions? Yes. Tim.
So I'm interested in the idea of dynamically rewriting the genome as you go along, because it strikes me that the cells already do that, but they do that with chromatin rather than with editing the DNA bases. So why go down the DNA bases route?
I think certainly chromatin is one of them. One of the ways by which you can turn things on and off. And then the question I have, though, is I'm not sure chromatin editing allows you to basically rewire new signals in new ways. So I'd really like to be able to have genetic circuits where I can connect, for example, promoters to new genes at will, and just by providing these kinds of new signals. And I'm not sure there's a way to actually do that just by silencing specific portions of the genome or being able to activate that. I'd love to talk to you about that, but I'm not sure that's actually possible. Because you can think about kind of this exponential search space if I can associate any promoter with any gene at will. That's what we'd like to be able to do with dynamic rewrites. Yeah.
So Ron, like you, we've been very focused on landing pads and so on, but hacks are also
Oh, yeah.
a real possibility. And I was wondering whether your group has looked at them and what your experience has been, and compare and contrast your
Yeah.
experience.
So hacks, the traditional ones are very difficult to work with. We try to do that with some of the earlier technology, and they weren't very stable. But there's some really exciting new technology. I think Alina from Pam Silver's group was here yesterday, so we actually had interactions with her. So I'm very excited about what she might be able to do with her hacks. I'd love to have hacks as well. Yep. Okay. Thank you.